So let us look at learning outcome for today's lesson. Can you see what's in the picture or not? This is what we're going to learn today. Okay, so firstly, investigate simple diffusion in the agar. And then also calculate surface area to volume ratio, illustrated, and also illustrate surface area to volume ratio will drop with an increasing size. And then investigate the effect of changing surface area to volume ratio on the diffusion using agar blocks of different sizes. So you can see in the pictures different sizes. Later on, I'll also show you a video where we change the shape because all the sizes here, all the shape are the same. Okay, they're all cube. So what if we make it more flat, like leaf like that? So how will it affect the rate of diffusion? Okay, so basically this is what we're going to cover for today's lesson. Okay, so how surface area to volume ratio changes with size of the organism. Now, if you talk about unicellular organism, it means organism that's made up of one cell only. Can you give example? Organism that's made up of one cell. Xi Xian.
Amoeba.
Amoeba, good. What else? Hei Wen. Hei Wen not here, I think. Yeah, Hei Wen's not here. Okay. Jing Er.
Teacher, can you hear
Yeah.
me?
Yes, I can.
Oh, bacteria.
Yes, very good. Bacteria. Bacteria is made of one cell, right? It's an organism itself, right? Okay. Give one more example. Andrea?
Paramecium.
Paramecium, yes. So we have got amoeba, paramecium, we have got bacteria, okay? So they are all unicellular organisms. That means the body is only made of one cell, and that one cell is already an organism. Now, for us, we are multicellular, right? We are made up of many, many cells. So other than animals, right? Animal. What else, what other organisms that are made up of many, many cells in one organism? John?
Plants.
Plants, yes, very good. So we have touched on Animalia kingdom. We have touched on Plantae kingdom. What other kingdom are made up of organism with many cells? Rebecca?
Fungi.
Again, fungi, yes, correct. Fungi. Okay, good. So we are actually focusing on today the differences in surface area to volume ratio for the organism, whether it's unicellular or multicellular, and how that affects our ability to obtain materials from the external environment and to actually remove waste to the external environment. Okay, right. Plants and animals are multicellular organisms. So therefore, because of multicellular, of course, they will be larger in their sizes because they got more cells, isn't it, compared to unicellular organism. Definitely, we are bigger. Okay, so what are the problems when our sizes become bigger and bigger? So let us look at the diagram to illustrate. Okay, so here you have got three cubes, A, B, and C, to represent three different organisms of different sizes. So A is only made of one cube. So let's say, for example, this one cube represents one cell. Okay, so this is a unicellular organism that you've mentioned. It could be amoeba. Okay, it could be bacterium. It could be paramecium. Okay, and then after that, we have got B, which is a multicellular organism. So in this organism, you can see that it's made up of, if one cube is one cell, so this will be made up of 8 cells, right? So say, for example, this is the fungi. Okay, fungi. And then C is even a bigger organism, which is multicellular. So let's say, for example, C is plant. Okay, it's plant, or animal also can, small animals. Okay, so C is made up of how many cells? 3. You go and count how many cells are there. Okay, all right. So what we want to see now is that we want to calculate, number 1, surface area of this organism. So we are actually looking at organism now as a whole. Okay, whether it's unicellular, okay, here, or multicellular here. Multicellular. So unicellular is small, whereas multicellular is bigger. Okay, so we're going to look at that. Now, so first we look at surface area and then followed by volume. So in the diagram, the surface area and volume has been calculated and given already. So let us just check whether it's correct or not. Now, if the site is 1 unit times 1 unit, it means this is 1, and this is 1. Okay, it can be 1 mm, 1 micrometer, it cannot be 1 nanometer. Okay, 1 micrometer can. Okay, so let's say 1 micrometer, 1 micrometer, 1 micrometer for the size of a cell. Right, so the surface will be altogether surface. What is that? Surface means surface area. We're talking about surface area. Okay, so each surface of a square is 1 times 1. So altogether you have got 4 squares to form the surface, not 4, sorry, 6. 6. So it's actually 1 times 6, so it's 6. Okay, then the volume is 1 times 1 times 1. Okay, so therefore it's 1 micrometer cube. So now the more important thing is this formula here, surface area to volume ratio. Okay, surface area to volume ratio is 6 divided by 1. Okay, surface area is 6, volume is 1, so 6 divided by 1 is 6. Okay, so this is calculation for the unicellular organism. Now let us look at B for fungi. Let me clear off first. So now this fungi is multicellular, so the site now is actually 2 times 2 times 2. So this is the length of the site of the organism already, made up of 8 cells. Okay, so the surface you calculate is actually 24. Right, surface is 4 times 6. Right, this surface here is 4. How is it 4? 2 times 2 is 4. Okay, altogether this whole structure has got 6 flat surfaces. So it's 4 times 6, so it's 24. Volume is 2 times 2 times 2, so it's 8. So therefore the surface area to volume ratio is 24 divided by 8, so it's equals to 3. Okay, so from 6 to 3, can you see there is a decrease in surface area to volume ratio? So later on, we will actually check how to understand this part. Okay, now you calculate, you get it, right? Look at the diagram. Can you get it directly without calculating? Actually, you can. Okay, so now we look at the third cube here, which is even bigger, representing bigger organism with even more cells. Right, so the site is now 3 times 3 times 3. Okay, so therefore the surface area is 3 times 3 times 6 flat surfaces, so it's 54. The volume is 3 times 3 times 3 is 27. So surface area to volume ratio is 56 over 27, so it's equals to 2. So again, you see from here to here, surface area to volume ratio drop even further. Okay, so from this calculation, we can say that as the size of organism increases, okay, the surface area to volume ratio will decrease. Now, this one we do by calculation, right? If you look at the diagram alone, can you reason it out or not? Okay. Now, in the first example here, which is a unicellular organism, one cell. Now, in this one cell got 6 surfaces, right? Because it's cube shape. It's represented by the cube. So all the 6 surfaces are facing the external environment, and hence all these 6 surfaces can be used for exchange of materials, because this organism, what does it need to take in from the external environment? Sufyan? What does it need to take from external environments?
Nutrients.
Yes, nutrients. Nutrients like carbohydrate, lipids, proteins, right? Other than nutrients, what else? Peiwen. Sorry, I keep on calling Peiwen. She's not here today. She's okay. Yun.
Oxygen.
Huh?
Water.
Can I hear you?
Oxygen and water.
Yes, very good. Oxygen and water. Yes, these are all very important molecules that our body need, right? Oxygen for respiration, water for hydrolysis, water for hydrating the cell, okay, and many other functions and nutrients. So these are the things that we need to keep on obtaining from external environment, okay? Now, what do we need to remove from the cell to the external environment? Daniel? Is my Daniel here?
Yes, teacher. Not sure.
Okay, think of yourself. Every day, what you need to remove from your body.
Oh, feces.
Yes. Yes, but the cell will not have feces. Okay, the
Oh,
cell will
sorry.
have. It's okay. Yes, feces. Feces is not waste actually. Feces is considered as undigested material, right? Now, what about the
Yes.
urine? Urine, you're removing urea, for example, which is actually a metabolic waste. Okay? You know the difference between your feces and your urea, right? Feces are those things that your body did not digest. When you consume the food, you did not digest it. So those are feces, okay? Whereas urea is a metabolic waste. That means, remember, metabolism is always represented by A plus B to form, where A plus B to form C plus D is considered as metabolic reaction. So all metabolisms in the body are represented by this kind of chemical equations. So the moment there's some products that your body do not need anymore, those metabolic products are considered as metabolic waste. Okay, for example, CO2 from respiration. After respiration, right, we produce a lot of CO2, but CO2, we do not need it. We have to remove it from our lungs, right? Whereas for bacteria, amoeba, and paramecium, they're unicellular. So very easily, the CO2 just diffuse out of the cell through the membrane. Okay, so we want to remove metabolic waste from the body. And also undigested material. Let's say, for example, this is an amoeba, right? They phagocytose some solid particles and digest it. Undigested material removed by exocytosis. Okay, yeah, okay. So these are showing we are very dependent on the external environment because we need to constantly exchange materials with the external environment. Okay, exchange. Exchange means you want to, meaning take in or release. Okay, take in from environment or release to the environment. Okay, so therefore this exchange of material requires it happens in our boundary of our body. Okay, so for unicellular organisms, very easy, just directly across the membrane. So in this case, if it's a cube, there are 6 surfaces that are directly exposed to external environment. So it's wonderful. Now, one step up. What if it is a multicellular organism like fungi? Now you examine one cube here, which represents one cell. Now, out of the 6 surfaces that it has, how many surfaces now are facing external environment? Chloe.
Teacher, can you repeat the question?
Now looking at these multicellular organisms, right, which is made up of, let's say, 8 cells here, that you can see in the diagram. Now, we go and examine one of the cells, which is the cube that I've circled. So for this particular cell, how many of its 6 surfaces are facing the external environment and hence able to exchange materials directly with external environments?
3.
3 only, right? So can you see the surface area in immediate contact with external environment has decreased already? Correct or not? So initially was 6, now it's 3 because the remaining of the 3 are facing the internal environment already. Correct? So this for all the cells here. Now, what about we go one step further up? So let's talk about human, multicellular. So if you talk about this cell here, right in the center, out of all the 6 surfaces, right, how many surfaces is facing external environment? Jia Ying.
1.
1 only. Can you see from 6 surfaces become 1 surface only? Only 1 surface is able to face external environment for this example. You're talking about this cell here, then it's 2. Right? If you're talking about this cell at the corner, then it's 3. So it's no longer 6. So can you see that as the organism increase in size, they increase in size because they are made up of more cells, multicellular. So therefore, the surface area that is able to be exposed to external environment actually drops already for every cell. We're talking about every cell per unit cell actually drops already. Okay. So therefore, how efficient is it now if the exchange of materials occurs immediately with external environment through the external surface, the surface facing external environment? How efficient it is for the cell in the center of this organism. So I'm talking about cell in the center, somewhere in the center there. How efficient is it for it to obtain nutrients and oxygen from outside and to remove the waste to outside? Is it as efficient as this cell here? Definitely it's not, because the cell is very far. The cell inside the body. Some cells in the body are totally not exposed to external environment. So therefore, to wait for diffusion of oxygen and glucose into this cell and to wait for the removal of urea from this cell to external environment is going to be quite slow and inefficient, isn't it? Slow, okay, inefficient. Why? Because the location of this cell is so far from the exchange surface. So the cells are far from exchange surfaces. Exchange surfaces are surfaces immediately in the external layer of the organism. Okay, your skin layer, for example. So the exchange is not efficient anymore. So how do we illustrate this? We illustrate this using this formula, surface area to volume ratio. So why do we do this? Is because surface area is what we are concerned about. That surface which allow exchange of materials to occur for this organism. So therefore, we are very concerned about surface area. We also talk about the volume because our internal environment of body is where we need to have got glucose and oxygen to be supplied to all the cells, because all the cells are immersed in the internal environment, which is the liquid. So we're talking about the entire volume of our body. Okay, so with this volume that I have for my body, do I have enough surface area to bring the materials into my body to supply to all the cells in this entire volume? So therefore, when I want to know the efficiency of exchange of materials for the organism, I will have to take into consideration both surface area, where exchange of materials occur, and volume is how big is my body now. Okay, which represents, volume represents the cells. The more cells you have, the more is the volume. Okay, so you can see from here that as the size of organism increases, definitely the surface area to volume ratio will actually decrease. It means the surface area will not increase as fast as the volume. So that will lead to an inefficiency of exchange of materials directly from the surface of the body of the organism. It's not sufficient anymore. Okay, so therefore, how do we adapt to overcome this problem? For us and for the plant, we use the so-called transport system. Okay, transport system is like you build a highway across the entire country. So you can send things through every part of the country very quickly using the highway. Okay, because diffusion directly alone is too slow already, right? You swim across the whole sea is too slow. Okay, so use a tunnel or whatever to transport things very quickly. So we use transport system and we use lungs, right? We use lungs to increase surface area so that we can take up oxygen and remove CO2 quickly. So you've got special organs and system to help us with this problem. Okay, whereas other organisms, they may not have this transport system and a lung system as well as us, because they are able to obtain whatever they need directly through diffusion because the sizes are small. Okay, so that's why you can see that as organisms get bigger and bigger, the body becomes more complex. There'll be more complex organ and system so that they can function efficiently even though they are big. Okay, so this is for you to understand very clearly the principle of this Y surface area to volume ratio. Okay, now, so we are concerned about doing experiment now to illustrate this surface area to volume ratio. Okay, so we can use a cube to represent the cell or organism. So this cube is very easily produced in the lab, like how you make agar at home that you eat. Okay, so you can make agar, and then this agar is also when you make the agar, you add a basic solution to it. Okay, can you give me example? What are the chemicals that make the solution becomes basic? Chloe.
Basic.
Yeah. Cepat.
Cannot think of any.
Cannot. Okay. Who wants to try? Yuhan, give me some chemicals as basic that you've learned before.
Sodium hydroxide.
Yeah, sodium hydroxide. If I don't have sodium hydroxide, I can use potassium hydroxide, right? Sodium carbonate also can, isn't it? Okay, sodium carbonate, but it's not so strong. So these two are stronger. So I will produce agar. You know how you make agar at home, right? You dissolve the agar in water when you boil it. And after that, I'm going to add the base into it. After that, I'm going to put it into a mold and let it set, right? After that, I can cut it out. Okay, I'm going to use ruler to measure and cut it out so to get the cube size that I want. So now I've got 3 different cube sizes. And then I also immerse this agar into this pH indicator phenolphthalein, all right? So to make it pink color. Now, what does phenolphthalein show? Is if you look at phenolphthalein, right? In different pH, it will give you different color. Now you look at these 3 cubes here, it's actually pink color. Pink color because when I prepare the agar, I've already added base into it, so it's actually basic. So it's basic, you can see that as long as it's basic, above pH 7 to 14, right? You can see the color is actually pink color. So if this kind of pink color should be pH above pH 9 lah. Okay, so that's how I prepared my agar for this experiment. So after that, the cubes are immersed into HCl solution for 10 minutes. So you can see 3 beakers here, right? And then I put the cubes into 3 separate beakers, which is made up of HCl, and I leave it there for 10 minutes. Now, during these 10 minutes, my HCl is going to diffuse into the agar because you know that diffusion will happen from region of higher to lower concentration, right? So therefore, my HCl is definitely going to diffuse into the agar cubes in this 10 minutes. So you know that when the HCl diffuses into the agar and it reacts with the HCl, okay? And going to react with sodium hydroxide that you already added into the agar. So it's going to neutralize, right? So it will make the agar becomes neutral, okay? So when the agar becomes neutral, the color will change from pink color all the way to maybe pH 7 or less than that. If it's less than pH 7, it becomes slightly acidic. Okay, if slightly acidic, then becomes white color. Actually, it's not white color, it's colorless. It becomes colorless. So we want to see, once we want to see where the HCl has diffused into, wherever it diffused into, that region will change color from pink to light pink, and that becomes colorless when you've got even more HCl than NaOH inside there. Okay, so it becomes colorless. So as an acid diffuses into the agar blocks, phenolphthalein will changes from pink to white color. It's actually colorless. Okay, so please write that into your notes, colorless. And you can also explain why. Okay. Now, so this is to show you how it will change. Now, in these 3 cubes, right, instead of using phenolphthalein, the scientists actually use bromo-thiamal blue indicator. It's also a pH indicator. It's either pink color or blue color. Okay, so let's look at the changes here. I think blue color is basic. Yes, that means, yeah, correct. Blue color is basic and then they put acid into it. Correct. Okay, now, you see what happened? After some time, and then after some time, can you see or not? So as more and more of the HCl diffuse into the agar, you can see that the region where the HCl diffuse into it becomes white color because pH has changes. Now I want to look at these 3 cubes, which represent 3 different organisms of different sizes. You can see the efficiency of diffusion now, right? For the first cube here, in 10 minutes, the oxygen from external environment can reach to the center of the cell here. Can you see or not? Because the entire cube is totally white color already. Whereas for the second cube here, you see in the center here, in this region here, this region, the oxygen cannot diffuse up to this region yet. Can you see? And then for even bigger size organism like this one here, is worse. You see or not? A huge volume of this organism or this cube, right, represented by this cube, a huge volume here in 10 minutes is unable to obtain the oxygen that's diffusing directly into the cube. So you can see that, imagine all the cells here, right, they're unable to obtain oxygen that's diffusing from the external environment yet within 10 minutes. So you can see the efficiency of exchange actually decreased when increased in size. Okay, so one thing is, now the rate of diffusion is the same for all of them, because if you measure the distance diffused, right, the distance diffused, can you see or not? And the distance diffused is actually the same because the kinetic energy of the molecule is the same, the temperature is the same. So therefore, the distance diffused in 10 minutes is actually the same for all the 3 cubes. But the percentage volume of the cube being diffused is different, right? Right? This percentage volume of the cube being diffused is 100%. Whereas this percentage, it could be maybe 70%. Okay, out of total volume, about 70% or 75% already diffused across. And whereas for this last one here, maybe only 20%. Out of 100% of the volume of the cube, only 20% of the volume cube has been diffused by the HCl. So this to show the efficiency of diffusion as the size of the organism becomes bigger and bigger, is actually decreased, right? So here there's a table, right? I have modified the table slightly to be aligned with the correct format of drawing the table. Yeah, I obtained the table from internet, so I have no time to change it. So I've already changed it yesterday. So it will be changed in your PowerPoint already, but not yet in your notes. So this is the way you need to write the format for table for A levels. Okay, the heading all the heading must be indicated very clearly what are you talking about, and all the units must be in the heading. So volume of cube, the unit is stroke. Stroke means followed by the unit. So the unit is cm cube. Area of cube, stroke, followed by cm square. Distance of diffusion, stroke, followed by distance is in cm. So this is the format to write the heading. Okay, I think I forgot to remove the cm here. Okay, there should not be any cm inside. So you see, once you remove all the unit, when you look at the table, it's very clean and neat and tidy. You can see the figure very quickly, right? Imagine compare if you write all the units and all the numbers inside, it's not going to be able to see very easily. So therefore, all units must be written at the heading rather than the body of the table. Okay, so that's the heading. This is the heading. And this is the body. Where's the body? This is the body. Okay, so this whole thing is the body of the table. So the head and the body. And this one unit in the body here is called a cell. It's called a cell. A cell in the table. One cell in the table. So all the cells, all the border of all the cells must be drawn with ruler and pencil, like this table here. Okay, you must use ruler and pencil to outline and separate all the cells with lines. Okay. Okay, so what you can see here is, again, you've got 3 cube sizes. This is the volume calculated in cm cube, and this area calculated. And this is the distance of diffusion after 10 minutes. You can see all of them are the same, right? Because I've shown you already, it's all of them the same. So in this case here, 0.5 cm. Okay. What about surface area to volume ratio? After you calculate, you can see that as the size of organism increases, okay, and the size increases, the surface area to volume ratio will actually decrease. Okay, so let's label it clearly. So this is for the size, and this is for the surface area to volume ratio. Okay, so the rate of diffusion is still the same. You can see now the rate of diffusion is how many cm the acid has moved into the agar in 1 minute. So you calculate, right? They're all the same because the distance is the same here. The only thing that's different is percentage of volume that's been diffused is different, right? So I'm going to use a next table to illustrate that. So as the size increase, diffusion rate in distance per minute is still the same, right? But what? So you look at this table. This table also I obtained from internet. So you can see these 3 sizes are different from the previous 3 sizes, slightly different. Okay, but still illustrate 3 different sizes. Now focus on this one now. Percentage volume of diffusion is different. Now they put plus and minus here because this is an experiment, right? This is an experiment. So they have repeatedly, maybe a class of 20 students did the experiment. So 20 students definitely have got different measurement. So they take an average of the measurement and they make a plus minus. Okay, so therefore you got this plus minus, which you call it as an error. Okay, an error of the experiment. So we got 36. So don't look at plus minus 6. We look at 36. Okay, 36% of the total volume diffused, 50% of total volume diffused, and 100% plus minus 2. Okay, 100% plus minus 2. Okay, plus, add the plus on top of the minus, is being diffused. So therefore, different sizes, even though the rate of diffusion is the same, but the percentage of volume being diffused will be different. The higher is the size, the percentage volume diffusion will actually drop. So there's an inefficiency for direct diffusion to obtain materials and to remove materials from the body of the organism. Okay? Okay, so cell has to be small for efficient diffusion of nutrients into the center of the cell. So if the organism becomes big, the cell is still small. So for organism becomes big, we will have got special system to help us to cope with this problem of unable to obtain nutrients and oxygen by direct diffusion alone, okay, from our external environment. Okay, so this is a very interesting video. I've changed the video already. Yesterday after viewing, I've changed the link of the video because this video is quite interesting. I think we have time to watch this video. Would you like to watch this video? Yes? Okay, let me try how I can do that. Okay, I will